Namaste. हमेशा खुश रहो बिटिया. I am JV Dr. Lily Trivedi, and today I am going to take the lecture of B.Sc AG students, B.Sc AG second sem students, and the lecture today is of the topic respiration. Now in respiration, today I am going to take only two parts, that is glycolysis and TCA cycle or Krebs cycle. Now let's start with what is type of respiration. Now what is respiration? The cellular oxidation or breakdown of carbohydrates into carbon dioxide and water, and the energy release is called respiration. It is a reverse process of photosynthesis. In respiration, the oxidation of various organic food substances like carbon dioxide, fat, protein, carbohydrate takes place, and among these, glucose is the commonest. Here you see, this is a glucose molecule and oxygen is taken in, and the final product we are getting carbon dioxide, water and energy, that is 686 kilocalories. Now there are two types of respiration, aerobic and anaerobic. In the aerobic respiration, the oxygen is there. In the presence of oxygen and complete utilization is there into carbon dioxide, water and energy. This is the most common occurrence type of respiration. The second is anaerobic respiration. It takes place in the absence of oxygen, where the respiratory substrate is incompletely oxidized. Now, the respiration is of rare occurrence and it is common in microorganisms like yeast. The glucose is converted into ethanol and carbon dioxide is released with less amount of energy, that is only 56 kilocalories. Now, the main mechanism of respiration involves glycolysis, aerobic breakdown of pyruvic acid, that is Krebs cycle, electron transport system, or also known as terminal oxidation or oxidative phosphorylation, and pentose phosphate pathway. Now, glycolysis or EMP pathway is common in both anaerobic and aerobic respiration. The overall glycolytic process is glucose is converted to pyruvic acid, 2 ATP is used, 2 NAD plus 4 ADP plus 2 molecules of phosphoric acid H3PO4. And finally we get pyruvic acid, 2 ADP, 2 NADH2 and 4 ATP. Thus there is a gain of 2 ATP molecules per hexose sugar molecule oxidized during this process. Besides these, 2 molecules of reduced coenzyme NADH2 are produced per molecule of hexose sugar in glycolysis. Now during aerobic respiration, these 2 NADH2 are oxidized to the electron transport chain to give 3 ATP molecules each. The 6 ATP molecules are formed. Now this is the process of glycolysis. Let me enlarge it. Okay. Now what happens here? The first process is the glycolysis takes place in absence of oxygen. Now what happens here is that the glucose molecule reacts with ATP in the presence of enzyme hexokinase, and hexokinase in the process of hexokinase, and they are converted to glucose 6-phosphate. Now glucose 6-phosphate is further converted into fructose 6-phosphate in the presence of phosphohexoisomerase. So the fructose plus ATP is converted into fructose 6 phosphate plus ADP. So there is an isomerization of glucose 6 phosphate into fructose 6 phosphate. Now this fructose 6 phosphate with a molecule of ATP is converted into fructose 1,6 biphosphate plus ADP. The fructose 1,6 phosphate is converted into 2-triose 3-phosphoglyceraldehyde and dihydroxy acetone phosphate, and this is in the presence of aldolase. Now this 3-phosphoglyceraldehyde reacts with phosphoric acid and forms 1,3-diphosphoglyceraldehyde, where the reaction is non-enzymatic. And this 1,3-diphosphoglyceraldehyde is oxidized to form 1,3-diphosphoglycerate in the presence of triphosphate dehydrogenase and the coenzyme NAD+. Now this NAD+ acts as a hydrogen acceptor and is reduced to NADH and H ion in the reaction. Now the 1,3-diphosphoglycerate reacts with ADP in the presence of phosphoglyceric acid, phosphoglyceric transferase to form 3-phosphoglyceric acid and ATP. So here we have 3-phosphoglycerate plus ATP. Now what happens to the 3-phosphoglycerate? It is isomerized into 2-phosphoglyceric acid in the presence of enzyme phosphoglyceromutase. Now this 2-phosphoglycerate is again converted into phosphoenol pyruvate plus water, and further we have in the presence of enolase. Now this 2-phosphoenol pyruvic acid reacts with ADP to form one molecule of pyruvate or pyruvic acid and plus one molecule of ATP, and the enzyme is pyruvate kinase. So this is the process of glycolysis both in aerobic and non-aerobic reaction. So this is, you can read this more clearly also in the book. Now we have another cycle. Before that, let me conclude the glycolysis process. There is a gain of 2 ATP molecules. 2 molecules of reduced coenzyme NADH is also produced, and aerobic respiration, these 2 molecules are oxidized through electron transport chain. Now after this we come to the glycolysis. Now what is glycolysis? It is the cycle which is taking place after the pyruvate acid is formed. Now the pyruvic acid produced in the glycolysis enters the Krebs cycle for further oxidation, and this Krebs cycle is also known as citric acid cycle of tricarboxylic acid, and this is an aerobic process which takes place in the mitochondria where the enzymes are present in matrix. So this is the Krebs cycle. Okay, now this is the Krebs cycle. Here is the pyruvate, and in the presence of pyruvate dehydrogenase, it is converted into acetyl coenzyme. Now this pyruvate dehydrogenase converts into acetyl coenzyme, and further it gives carbon dioxide and NADH2. So it is this process which releases carbon dioxide. Now we have acetyl coenzyme which condenses with oxaloacetate acid in the presence of condensing enzyme and the water molecules to form citric acid. So coenzyme becomes free. Now acetyl coenzyme plus oxaloacetic acid in the presence of a condensing enzyme plus water converts into citric acid plus coenzyme A. Now citric acid is dehydrated in the presence of aconitase to form cis-aconitic acid. The cis-aconitic acid reacts with one molecule of water to form cis-aconitic acid. Now what is the enzyme? The enzyme is aconitase. Now the cis-aconitic acid plus one molecule of water will form isocitric acid. Now isocitric acid is oxidized to form oxalosuccinate acid in the presence of isocitric dehydrogenase. So here NADP is also reduced to NADPH2 in the reaction. Now the oxalosuccinate acid is decarboxylated in the presence of oxalosuccinic dehydrogenase to form alpha ketoglutaric acid, and the second molecule of carbon dioxide is released. Now what happens here? What is formed? Alpha ketoglutaric acid. So now this reacts with coenzyme A and NADP in presence of alpha ketoglutaric acid dehydrogenase complex, and it is oxidatively decarboxylated to form succinyl coenzyme A, and the third molecule of carbon dioxide is released. So NAD is reduced. The succinyl coenzyme A reacts with water to form succinic acid, and the coenzyme A becomes free, and one molecule of guanosine diphosphate is phosphorylated in the presence of inorganic phosphate to form one molecule of guanosine triphosphate. So this reacts with ADP to form one molecule of ATP and again guanosine diphosphate. Now we have succinic acid. Now what happens to this succinic acid? It is oxidized to fumaric acid in the presence of succinic dehydrogenase, and coenzyme FAD is reduced to FADH2. Now one molecule of water is added to fumaric acid in presence of the enzyme fumarase to form malic acid. Now after this, this fumaric acid now is converted into malic acid in the presence of water and fumarase. Now the last step is malic acid is oxidized to oxaloacetic acid in the presence of malic dehydrogenase and one molecule of coenzyme, that is NAD is converted to NADH2. So this is what you have seen in the cycle, and the last we have the malic dehydrogenase which converts it into the malic acid. So this whole is the Krebs cycle which we have studied. So students, in the further lecture, I will be taking the next process. So today we have studied glycolysis and Krebs cycle. So I thank you. If you have any query, please write in the comment box and I will resolve it soon. This session was powered by Digital 2.0 JAYOTI VIDYAPEETH Women's University. So thank you very much. Namaste.